Hey guys, how you doing? So welcome to the second video of chapter 11. We are going to look at meiosis in this particular video, and we're going to focus on the kind of ins and outs of that process and why it is different from mitosis. Okay, all right, let's jump into it. Okay, so meiosis. Now, unlike mitosis, meiosis has 8 stages, twice the number of stages that mitosis has, and it is a little bit more complicated, but it requires you to understand how mitosis works so that you can understand meiosis. Okay, now both mitosis and meiosis will have the interphase, so I'm not really kind of including that in, but you know, quite conveniently or inconveniently, depending on how you want to look at it, meiosis has a very similar naming of stages or phases as mitosis. Okay, so the first one is called prophase 1. Yeah, that I looking thing is a 1. Yeah, it's just the font makes it look like an I, but it's prophase 1, that is metaphase 1, anaphase 1, and then telophase 1 and cytokinesis. Yeah, now notice how I've sort of lumped telophase 1 and cytokinesis together. Yeah, and even though technically they should be kind of their own stages. Okay, and the cycle then repeats again, and so you get prophase 2, and then you can probably guess what the next ones are: metaphase 2, anaphase 2, and then telophase 2 and cytokinesis again. Okay, now here is just a bit of an overview, and you will, if you pick out carefully enough, see elements of mitosis in there as well. Okay, so the easiest way to distinguish between the phases and whether they are a mitosis phase or whether they are a meiosis phase is that the numbers after the phase will always indicate meiosis. Yeah, so if you see just prophase on its own without a number, that will tell you that that prophase is referring to mitosis. If it has prophase 1 next to it, it is referring to meiosis. Okay, but here's just a kind of bit of an overview that you can see there. Now, you will notice that throughout the first round of meiosis, that the cell doesn't really split, yeah, and then it kind of, similar to mitosis, it kind of retains its kind of whole form, and then it will split in half here, and then each one will undergo their own particular specialized phases, and then they will split up again. Now the result is you've got 4 times the number of daughter cells. Yeah, so you've got 4 individual daughter cells as opposed to only 2 that you would normally find in mitosis. Okay, but each step looks slightly different. Okay, so we're going to run through each of these phases, and you're going to have to bear with me because there's going to be quite a bit of intricate detail, and it might be good for you guys to refresh your memory on how mitosis works if you are not sure, because some of the details in mitosis I'm going to kind of gloss over a little bit here to emphasize some of the new stuff. Okay, all right, meiosis round 1. Let's go. Prophase 1. In the first prophase, it is very similar to prophase in mitosis. In the nucleus, you have the chromatins shortening and condensing to form chromatids, and then they will become visible. The chromatids will then form your chromosomes. Okay. Now, in the cytosol, which is the rest of the cell, the centrioles, which are the organelles that are responsible for developing the microtubules, will begin to form your spindle network. Very similar to mitosis, same kind of form. The nuclear membrane will then dissolve, and you have, once again, a very similar occurrence. Yeah, so there's your centriole. There's the centriole creating the spindle network and then moving to opposite sides so that they are creating your spindle fiber network there. And then the nucleus membrane dissolves and the chromosomes will begin to line up towards the equator as it transitions into metaphase. But wait, there's more. Okay, so still in prophase 1, there is a particular stage that occurs, and what will happen is the matching pairs of chromosomes, which are homologous, will then line up, and they will do a special thing where they exchange DNA with one another. This is where chromosomes in your own cell, the matching pairs, yeah, will exchange their own DNA to be slightly different. Okay, this particular process is called crossing over, and then what you have is that the DNA from the same locations of the chromosomes will then exchange, and the resulting chromosome is a recombination of the previous ones. Yeah, so this whole kind of phase is called crossing over and recombination, because the DNA is now becoming different from what it was previously. Okay, the DNA of each chromatid will be different from each other chromatid, with the exception of the male sex chromosomes, and that's because in the male sex chromosomes you have an X and a Y sex chromosome, and they can't exchange because they don't have the same locations. Okay, so where if it's an X chromosome and an X chromosome, they'll be able to do recombination. Okay, here's a diagram of what that looks like. Okay, so you can see there, here is a matching chromosome in blue, yeah, and here is the kind of the matching pair in red. And what will happen is their tail ends will sort of overlap each other. That's crossing over right there. Okay, the tail ends cross over, and then they will kind of detach and adopt the other side's section of the chromosomes. Yeah, remember, these chromosomes are only different in terms of the amount of detail. Yeah, this particular section might have, you know, genes for particular traits, and then the same section will occur in both. But what happens is they then exchange the information, so you get a slight different recombination or variation. Yeah, so you know, rather than having gene A, gene B, and gene C capital, you now have gene A, gene B lowercase, and gene C lowercase, and then the other variants as well. Yeah, so that's crossing over and recombination. It's an excellent way in which the chromosomes will have a little bit of variation in them, which then allows them to be different even more. Okay, later on, when these chromosomes split, they will split down the middle here, just like in mitosis, and you will notice that then this strand here is all blue, this strand is blue at the top and then red in the bottom, this strand is red at the top and then blue at the bottom, and then this strand will be all red, and so that each of these strands will go into a different sperm or a different egg and look different. Yeah, that's the recombination and the crossing over occurring, and it happens in prophase 1. Okay, now metaphase 1. After that happens, the chromosomes will then line up at the equator, which is the metaphase plate, and attach to the spindle fiber, similar to mitosis. However, the chromosomes will lie horizontal so that the chromatid faces away. The nucleus dissolves. The difference here is that the chromosomes line up in pairs rather than on their own. Okay, so that's what they look like. Yeah, rather than one lining up at the equator as you saw previously, they will line up in pairs. So there's one there, there's one there, there's one there, and there's one there, and that's your metaphase, the equator. Okay. How is that different from mitosis? Well, in mitosis, they line up in singular, yeah, along there, and then each one will be split in half right down this middle and move to their respective sides. However, in meiosis, they line up in pairs, and what you'll notice is that when the pairs do split up, this whole chromosome will move to one half of the cell, this whole chromosome will move to the other half of the cell, and so what you get is this half of the cell will have 23 whole chromosomes. Okay, 23 whole chromosomes. This one here will have 23 whole chromosomes, or 23 pairs, right? And so each one has basically become a diploid cell on its own. Right? The cell will then transition to anaphase 1, where the centromere divides and pulls each double-stranded chromosome to their respective sides. Yeah, similar to mitosis again, spindle fibers will then shorten. The homologous pairs of chromosomes will move to the opposite ends of the cell, not the chromatids, but the chromosomes. Okay, each half does not have the same DNA as the other half. Remember, because of crossing over and recombination, the DNA is basically all mixed up and they all look slightly different. Yeah, so you have a slight variation in the DNA. Yeah, in mitosis, it was one half going to one end and the other half going to the other end. In this case here, the whole chromosome moves to the opposite sides, and then it moves again to the opposite sides there. Okay, so this half here is now genetically different from this half here because of the crossing over recombination factor. Okay, telophase and cytokinesis is pretty much identical. Yeah, the two sets of chromosomes will form tight clusters again, and a new nuclear envelope will form around them to create two new nuclei in the cell. The spindle fiber then disassembles. However, each half does not have the same DNA, and the chromosomes will not unwind. The reason is because they're going to go into round 2, so they are not going to unwind and turn back into chromatins and become less visible. They're going to stay visible and wound up as chromosomes because they're going to go back into into round 2. Yeah. In cytokinesis, the membrane then pinches off, called cleavage, and divides the cell into 2. So you've got a very similar occurrence. The only difference here is that the chromosomes don't unwind, and then each half are not genetically identical. So that brings us to meiosis round 2. Meiosis round 2 is basically identical to mitosis, except it all happens with only half the number of chromosomes. Yeah, so mitosis happens with 46 chromosomes. Meiosis will happen with 23 chromosomes, right? And then each one will split into halves, right? So in prophase 2, the chromosomes condense and become visible, and the spindle fibers will form. The metaphase 2, chromosomes will then line up horizontally at the equator, and the nucleus will then dissolve again. In anaphase, the spindle fibers will shorten, and each chromatid moves towards opposite ends of the cell. And in telophase, a new nuclear envelope will form over each half, and the spindle fibers will disappear. The chromosomes now uncoil and become invisible. Right? So in cytokinesis, the plasma membrane then pinches off, and the parent cell divides into 2 daughter cells. So you can see there, this happens in summary there, and you can see the progression as it makes its way across. You've got your crossing over and recombination there, the mixing of DNA, chromosomes lining up along the metaphase plate in pairs and not in singles, and then each one is being split off. The cell splits up, divides into 2, and then restarts the meiosis round 2 phase again, and then in the end, what you've got is half the amount of DNA in 4 cells. Okay, so that's kind of how meiosis works, right? Now, in the end, what you've got is you've got one diploid cell or a germ cell producing 4 haploid gametes. You've got 4 times the number of cell produced there. Each haploid cell is genetically different from the other 3 because of that crossing over and recombination. Remember, yeah, you've started off with these kind of chromosomes there, the red and the blue, and then they cross over, that crossing over, recombining to form a recombination, and then during meiosis round 2, these ones will split into their individual chromatids, which is there, and you've got each one there looking different from each other one. So if I was to sort of put it plainly, you could say that this strand here from this chromatid will go into a sperm, this one here will go into a different sperm, this one here will go into a different sperm again, and this one here will go into a different sperm again, right? So this happens, now all this kind of recombination happens with 1 pair of chromosomes, yeah? In humans, we have 23 pairs of chromosomes, yeah? So if you do this with every single pair, you're going to end up with genetically vastly different sperm or egg from the parents, okay, which is where the variation comes from, right? And you can do the maths around that to calculate exactly what are the chances of being identical and what are the chances of being different from either parent, yeah? So the diploid number of chromosomes 2n, suppose you have 4 chromosomes in your body as opposed to 46. The diploid number 2n equals to 4 means that n equals to 2, yeah? And what you do is, this variation is a 1 in 4 chance of differences, yeah? And then you can square that because of n equals to 2. So 1 in 4 to the power of 2 will give you 1 in 16 of having identical combinations. So that is the off chance. There is only a 1 in 6 chance that you will be the same as your dad or your mom, in terms of the sperm and the egg, right? If you have 4 chromosomes, right? If you have 6, 2n equals to 6, n equals to 3, that means it is 1/4 to the power of 3 which is 1 in 64 chance. So you can see there, with every rise in chromosomes, you have a drastically different or increased number of variants, right? This is a little bit of mathematics. I don't expect you guys to sort of know this table and how it works, but for those of you who are interested in understanding what the possibilities are, right? If you have only 10 chromosomes, there is only a 1 in 1024 chance that the sperm produced will be identical to the father, right? Or the egg produced will be identical to the mother. Now, if we have 46 chromosomes n to the 23, then you've got a factor of 1/4 to the power of 23, which is why genetically sperm are different from every other sperm. There is almost zero chance that the sperm will be the same as another sperm next to it, unless for some reason there is that kind of off chance there, right? The same goes for eggs, right? Now, for the eggs and sperm, production of sperm, or what we call spermatogenesis, spermato for the spermatozoa, and then the genesis meaning birth, differs from the production of eggs, which is called oogenesis. Okay, for sperm, one spermatocyte will form 4 sperm cells. Okay, so meiosis will produce 4 sperm cells from one spermatocyte. However, for eggs, what happens is the oocyte will produce 1 ovum only, and it will then produce 3 what we call polar bodies. Now, the polar bodies are basically useless. Most of the nutrients gets diverted to the ovum or the egg, and so there's nothing left in the polar bodies, and they just disintegrate or just become incorporated as part of the egg, right? So that's what it looks like. This is mitosis. This is meiosis at work, yeah, producing your sperm. There is your spermatozoa, or spermatocyte, splitting into 2 in meiosis round 1, and splits into 2 again into meiosis round 2 to form your 4 sperm. In your oogenesis, or your egg formation, meiosis round 1 will split, and then what will happen is that egg loses most of its nutrients to this one here, which will then split again, losing most of its nutrients, so that in the end you've got 1 ovum, and then you've got these kind of polar bodies that do absolutely nothing, okay? So that the egg retains all of its nutrients and doesn't kind of lose its size as a result, which is really important for females because that is the primary cell in which the zygote takes place, okay, or forms, right? So here's a summary between mitosis and meiosis. Mitosis occurs mostly in your body cells, whereas meiosis will only occur in the germ cells in the ovaries or the testes. The function of mitosis is for growth, increasing cell number and repair. The function for meiosis is the production of the gametes, the egg and the sperm. DNA is still replicated in the interphase, yeah, so you still have 92 chromosomes at the start of the mitosis phase and the start of the meiosis phase. The number of divisions, however, is different. Meiosis will undergo 2 rounds of divisions. Mitosis will only undergo 1. And there is a matching or pair at the synapses, which allows them to divide during the end of meiosis round 1. There is no matching of pairs or pairing of matching chromosomes in mitosis. Crossing over will occur in meiosis, but not in mitosis. If it did, you would have a lot of problems because every time your body tries to repair itself, it creates a different cell from what it was before, which is not ideal, right? And in the end result, you've got 4 haploid cells, none of them that are identical to each other in meiosis, but you've got 2 daughter cells that are exactly identical to the parent cell in mitosis. So you can sort of see that the summary for mitosis serves a different function: growth, increase in cell number, and repair, so you want many copies of the same thing. However, meiosis is specifically for the purpose of sexual reproduction, which is aimed for genetic variation, and so once again, you have a very different process that allows you to produce genetic variation. Okay, so that is a summary of meiosis. I hope you guys have learned something there, and if there's anything that you're not sure about, I would strongly encourage you guys to make sure that you kind of go over it again to make sure that you are ready and understanding of this process, okay? Because it can get really confusing if you don't spend the time to wrap your head around it, okay? I'll see you guys later.